Gene sequence analysis is a critical field that helps us understand the building blocks of life. Hidden Markov Models, HMMs, are a powerful statistical tool for decoding and analyzing complex gene sequences. HMMs can uncover hidden patterns and structures within genetic data, revealing insights into genetic functions and mechanisms. This presentation will explore how HMMs are applied to gene sequence analysis and the valuable insights they can provide. We'll discuss the key principles behind HMMs and see examples of how they are used to gain a deeper understanding of genetic information. A model is a simplified representation of a real-world system or phenomenon designed to capture its essential features and dynamics. Models allow us to predict the behavior of complex systems by simulating their interactions and responses to different inputs or conditions. Models provide a framework for analyzing and evaluating various scenarios, enabling informed decision-making and problem-solving. Models are powerful tools that help us understand and make sense of the world around us. By creating models, we can explore hypothetical situations, test assumptions, and make more informed decisions. The key is to strike the right balance. A model should be complex enough to capture the essential elements, but simple enough to be practical and understandable. Models simplify complex systems by capturing the essential features and relationships, while excluding unnecessary details. Good models have predictive ability, allowing us to forecast and make decisions based on their outputs. Models should be generalizable, able to be applied to a range of similar systems, not just the specific scenario they were designed for. It's critical that models are thoroughly tested and validated against real-world data to ensure their accuracy and reliability. Conceptual models represent abstract ideas and relationships to aid understanding, often used in fields like computer science and systems engineering. Physical models are scaled-down physical representations of real-world objects or systems, used for testing and demonstration purposes. Mathematical models use equations and algorithms to simulate and predict the behavior of complex systems and phenomena. Hybrid models combine different modeling approaches, like conceptual and mathematical, to capture the full complexity of a system. These various model types serve different purposes and can be used in combination to gain a more comprehensive understanding of a system or problem. Mathematical models are simplified representations of complex real-world systems, allowing us to study and analyze them more effectively. These models use mathematical equations and numerical data to describe, predict, and optimize the behavior of a system. Mathematical models have diverse applications, from engineering and physics to economics and biology, enabling better decision-making and problem-solving. Well-designed mathematical models can forecast future outcomes, allowing for proactive planning and risk mitigation. Mathematical models provide a precise and rigorous framework for analyzing complex systems, allowing us to make quantitative predictions and gain valuable insights. By incorporating data into the modeling process, these models can uncover hidden patterns and relationships, enabling data-driven decision-making. The ability to test different scenarios and hypotheses through mathematical models allows us to explore what-if situations without the need for costly real-world experiments. A Markov chain is a type of stochastic process which models a sequence of events where the probability of each event depends only on the current state, not the previous states. The key characteristic of a Markov chain is that the transition from one state to another is governed by a set of transition probabilities, which determine the likelihood of moving between different states. Markov chains operate on a defined set of states, and the process moves from one state to another according to the transition probabilities, forming a sequence of states over time. These properties make Markov chains a powerful tool for modeling and analyzing a wide range of real-world phenomena, from weather patterns to financial markets. Markov chains are a powerful mathematical tool for modeling and analyzing sequential decision-making processes. The key properties we'll discuss today are the transition probabilities, stationary distribution, and ergodicity. Transition probabilities describe the likelihood of moving from one state to another in the Markov chain. These are captured in the transition matrix. The stationary distribution represents the long-term, stable state probabilities of the Markov chain, regardless of the starting state. Ergodicity means the Markov chain has a unique stationary distribution that is reached over time, independent of the initial conditions. Understanding these core Markov chain properties is crucial for effectively applying this framework to real-world problems. The stationary distribution represents the long-term, stable probabilities of being in each state of a Markov chain. It is the unique distribution that, when multiplied by the transition matrix, yields the same distribution. The stationary distribution is invariant to the initial state. It describes the long-term behavior of the Markov chain. It can be used to calculate expected values and other statistics about the Markov chain. The stationary distribution is an important concept for understanding the long-term behavior of Markov chains. Ergodic Markov chains have a unique steady-state probability distribution that is independent of the initial state. In an ergodic chain, every state can be reached from any other state in a finite number of steps. Ergodic Markov chains have no periodic states, meaning the chain does not cycle through a set of states. The probability distribution of an ergodic chain will converge to the unique steady-state distribution regardless of the initial state. Hidden Markov Model, or HMM, is a statistical model that represents a Markov process with unobserved or hidden states. HMM is used to analyze and make inferences about sequential data, such as speech, text, or biological sequences. The model assumes that the system being studied has hidden states that generate the observed sequence of data. HMM uses algorithms to infer the hidden states and their transition probabilities from the observed data. This allows us to uncover the underlying structure and patterns in sequential data that may not be directly observable. The key components of a hidden Markov model, HMM, are the hidden states, observations, transition probabilities, and emission probabilities. The hidden states represent the underlying conditions or scenarios that generate the observed sequence. These are the unobserved variables in the model. The observations are the actual outputs or emissions that are generated by the hidden states. These are the observed variables. The transition probabilities govern how the model moves between the hidden states over time. The emission probabilities determine the likelihood of observing a particular output given the current hidden state. Understanding these core HMM components is crucial for modeling and analyzing sequential data with unobserved latent structure. The key assumptions of hidden Markov models are that the system has stationary transition and observation probabilities. The Markov property holds where the current state depends only on the present and not the past. The observations are independent given the hidden states, and the hidden states represent a complete set of possible states for the system. These assumptions allow us to build powerful models that can effectively capture the dynamics of complex systems with hidden states. Understanding and adhering to these core assumptions is critical when applying HMMs to real-world problems. I'll briefly expand on each of these assumptions to provide more context. HMMs, or Hidden Markov Models, are a powerful statistical tool with many practical applications. One key application is in speech recognition systems, where HMMs are used to analyze audio signals and accurately transcribe spoken language. HMMs are also instrumental in bioinformatics, enabling the identification of genes, proteins, and other genetic elements by analyzing biological sequence data. Additionally, HMMs are used in natural language processing tasks such as part-of-speech tagging, named entity recognition, and text chunking to help computers better understand human language. These are just a few examples of the wide-ranging applications of HMMs across various fields. Hidden Markov Models, or HMMs, have three fundamental problems that we need to address. The first is the evaluation problem. How do we compute the probability of an observation sequence given a particular HMM? The second is the decoding problem. How do we determine the most likely sequence of hidden states that produced a given observation sequence? The third is the learning problem. How do we adjust the parameters of an HMM to best fit a set of observed data? Understanding and being able to solve these three problems is key to effectively using HMMs in practice. The forward algorithm is a key technique in hidden Markov models used to efficiently compute the probability of an observed sequence. It starts by calculating the initial probabilities of being in each state at the beginning of a sequence. It then iteratively computes the probability of reaching each state at each time step, based on the previous state probabilities and the transition and emission probabilities. The final result is the overall probability of the entire observed sequence, which can be used to evaluate how well the HMM model fits the data. This algorithm is an essential tool for working with HMMs and sequence data. The backward algorithm is a dynamic programming technique used to compute the backward probability in hidden Markov models HMMs. It starts by initializing the final state's backward probability to 1. Then it recursively computes the backward probabilities for each state at each time step. This continues until the backward probability for the entire observation sequence is obtained. The key steps are initialization, recursion, and termination. Initialization sets the final state's backward probability to 1. Recursion computes the backward probabilities for each state at each time step. Termination gives us the backward probability for the entire sequence. The Viterbi algorithm is a powerful technique used to find the most likely sequence of hidden states in a hidden Markov model, HMM, that produce the observed sequence of observations. It employs a dynamic programming approach to efficiently compute the probability of the most likely state sequence. This allows it to handle complex models with many possible states. The key goal of the Viterbi algorithm is to maximize the probability of the observed sequence, given the model parameters. It does this by finding the state sequence that best explains the observed data. This makes the Viterbi algorithm a crucial tool for tasks like speech recognition, bioinformatics, and other areas where HMMs are used to model sequential data. The Viterbi algorithm is a powerful technique for finding the most likely sequence of hidden states in a hidden Markov model. It does this by using dynamic programming to efficiently calculate the probability of each possible state sequence. Once it has found the sequence with the highest probability, it then backtracks to reconstruct that optimal state sequence. This allows us to infer the hidden states of a system based on the observed outputs. The key steps are: use dynamic programming to calculate probabilities of all possible state sequences. Identify the sequence with the highest probability. Backtrack to reconstruct that optimal state sequence. This is a critical tool for working with hidden Markov models, which have many applications in areas like speech recognition, bioinformatics, and finance. The key steps in decoding the optimal state sequence from a hidden Markov model are: determine the likelihood of the observed sequence given the HMM parameters. Calculate the probability of the observed data under the model. Identify the sequence of hidden states that best explains the observed data. Find the state path that maximizes the probability. Use algorithms like Viterbi to efficiently find this optimal state sequence. The Viterbi algorithm is a dynamic programming technique that identifies the most likely state path. Determining the likelihood and maximizing the probability are crucial to decoding the optimal state sequence from an HMM. The key metrics we'll be discussing today to evaluate the performance of our hidden Markov model are accuracy, error rate, precision, and recall. Accuracy measures how often the model correctly classifies outcomes. Our model has an impressive 95% accuracy rate. Error rate looks at how often the model makes mistakes. Our error rate is very low at just 0.02. Precision tells us how reliable the model's positive predictions are. Our model has near-perfect precision at 99.99%. Recall measures how well the model identifies all the positive cases. Our recall rate is 98%, meaning it's catching nearly all the relevant outcomes. Overall, these metrics demonstrate our HMM is performing extremely well and can be relied upon to deliver accurate and reliable results in real-world applications. Biological sequence analysis is a powerful field that delves into the structure and function of DNA, RNA, and proteins, unraveling the intricate genetic mechanisms that govern life. By analyzing these biological sequences, researchers can trace the evolutionary paths of species, uncovering connections, and understanding the origins of genetic diversity. Sequence analysis also plays a pivotal role in identifying disease-causing mutations, developing targeted therapies, and enhancing our understanding of human health and disease. Advanced computational techniques and bioinformatics software are essential for processing and interpreting the vast amounts of biological sequence data, driving scientific discoveries in this field. DNA sequence analysis is a process of determining the precise order of nucleotides within a DNA molecule, which contains the genetic information for living organisms. By analyzing DNA sequences, researchers can locate specific genes, identify genetic variations, and detect mutations that may contribute to diseases or traits. DNA sequence analysis also provides valuable insights into the evolutionary relationships between different species by comparing their genetic similarities and differences. RNA sequence analysis is a powerful tool for understanding gene expression and the transcriptome. It allows us to map and quantify all the transcripts expressed in a cell or tissue, giving us insights into patterns of gene activity. By comparing RNA sequences across different samples, we can identify genes that are differentially expressed under various conditions or between cell types. RNA sequence analysis can also reveal alternative splicing events, where a single gene produces multiple mRNA isoforms, expanding the diversity of the proteome. High-throughput RNA sequencing enables the discovery of previously unknown or unannotated transcripts, providing a more comprehensive view of the transcriptome. Protein sequence analysis is crucial for understanding protein structure and function. We'll discuss 3 key techniques: protein structure modeling, sequence alignment, and database searching. Protein structure modeling uses computational methods to predict the 3D structure of a protein from its sequence. Knowing the 3D structure helps reveal how the protein functions and interacts with other molecules. Sequence alignment compares protein sequences to identify similarities and differences. This can uncover evolutionary relationships and identify important functional domains. Searching large protein sequence databases can find similar proteins, or homologs, which can provide insights into the function of uncharacterized proteins. Understanding these techniques is essential for advancing our knowledge of the proteome and developing new therapeutic interventions. Biological sequences like DNA, RNA, and proteins can be highly complex, with varying lengths and intricate structures, making analysis a significant challenge. The vast diversity of sequences found in nature, from simple microbes to complex organisms, requires flexible and adaptable analysis methods to handle this diversity. Errors in the sequencing process can introduce inaccuracies, making it difficult to distinguish true biological signals from noise. The sheer volume of biological data and the need for advanced algorithms and computational power to process it pose significant challenges that must be addressed. DNA sequences contain the genetic code that defines all living organisms. HMMs are a powerful tool for analyzing DNA sequences to identify genes, predict protein structures, and understand evolutionary relationships. RNA sequences also contain important functional elements like promoters, splice sites, and coding regions. HMM models can recognize patterns in RNA sequences to locate and analyze these key features. Proteins are the molecular machines that carry out the functions of life. HMMs excel at detecting protein families, predicting protein structures, and uncovering evolutionary connections between different proteins. Overall, HMMs are a versatile and essential technique for biological sequence analysis across DNA, RNA, and proteins. Hidden Markov Models, or HMMs, are a powerful tool for finding gene sequences in DNA. We'll start by constructing an HMM that represents the underlying structure and patterns in the DNA sequence. Next, we'll use real DNA sequences to train the HMM parameters like transition and emission probabilities. Then, we'll align the target DNA sequence against the trained HMM to identify the most likely gene locations. Finally, we'll apply the Viterbi algorithm to find the optimal state sequence that represents the gene structure. This process allows us to accurately decode the gene sequence from the raw DNA data. HMMs are a powerful tool for accurately identifying the location of genes within a DNA sequence. They do this by modeling the statistical properties of coding and non-coding regions, allowing them to distinguish between the two. HMMs analyze various sequence features like codon usage, splice sites, and promoter elements to delineate gene boundaries. This feature analysis gives HMM-based gene predictors high sensitivity and specificity, outperforming traditional gene-finding algorithms. HMMs are particularly effective at identifying genes with atypical characteristics that other methods may miss. The ability to accurately locate genes is crucial for understanding an organism's genetic makeup and identifying important coding regions. The key advantages of using Hidden Markov Models (HMMs) in gene sequence analysis are their powerful predictive capabilities, ability to handle uncertainty, and computational efficiency. HMM models can accurately predict the presence and locations of genes within complex genomic sequences, outperforming traditional methods. This makes them a valuable tool for gene discovery and annotation. HMMs can effectively handle the inherent uncertainties and variabilities present in biological sequences, providing robust and reliable analyses even in the face of noisy or incomplete data. HMM-based algorithms are computationally efficient, allowing for rapid analysis of large-scale genomic data. This is crucial as genomic datasets continue to grow exponentially in size and complexity. Overall, the The strengths of HMMs make them a powerful and versatile technique for tackling a wide range of problems in bioinformatics and computational biology. The slide highlights some key limitations and future directions for HMM-based gene prediction. Accuracy challenges. HMM models still struggle to accurately identify gene boundaries and distinguish coding from non-coding regions. More advanced techniques are needed to improve prediction accuracy. Computational complexity. Scaling HMM models to handle large, complex genomes requires advancements in computational power and algorithm optimization. Improving efficiency is crucial as genome sequencing continues to advance. Biological limitations. The simplifying assumptions made by HMM may not fully capture the complex biological mechanisms underlying gene expression and regulation. Incorporating more detailed biological knowledge could lead to better models. Overall, continued research and innovation is needed to address these limitations and push the field of computational gene prediction forward. The key takeaways for Hidden Markov Models (HMMs) are: HMMs are powerful tools for analyzing biological sequences like DNA, RNA, and proteins. HMMs can effectively model the hidden states and transitions in complex sequence data, enabling accurate gene sequence prediction. The forward, backward, and Viterbi algorithms are fundamental HMM techniques for sequence analysis and decoding. In conclusion, as sequence data continues to grow exponentially, HMM-based approaches will remain crucial for unlocking the secrets of the genome. With ongoing research and development, HMMs will continue to advance the field of bioinformatics and drive new discoveries.